Hello everyone, I'm Ning from the Hong Kong University of Science and Technology. Today I will spend half an hour time introducing the study we have done in Wuhan hospitals about the coronavirus during COVID-19 outbreak. So this is going to take 30 minutes. The topic of the study is talking about aerodynamic characteristics and RNA concentration of airborne SARS-CoV-2. This is a very, very hot topic, and I hope the information contained in this study will be useful for the reference of different places who is combating on the coronavirus at the moment. This is a joint effort from many universities and institutes. I will give a detailed explanation about the research team a little later on. This study has been preprint on the IXIV website, so you can feel free to download the paper. Now, a little bit of background about the study. This is exactly the moment, Saturday, April 4, 2020. The cases of COVID-19 global-wise has exceeded 1 million, and including over 200 countries at the moment. So this is a very, very serious issue. And this study is looking into how this coronavirus can potentially be transmitted in aerosol form, which is a very dangerous form. This is actually the website that you can go and download our study and see how these study results can look like, and also how we interpret the results and implications to protect ourselves as a general public and also the medical staff. After this paper was out on March 10, 2020, we have received many news reports. If you can see from the metrics, over 1,000 news articles have reported the study. That means really important subject on a global scale now. And you can see many important news outlets have seen the important information from the study. More importantly, and on April 1st, the National Academy of Science and in the US, they have formulated an important panel report to the US government talking about the rapid expert consultation on the possibility of bioaerosol spread of this virus for COVID-19. And in this important letter to address to Dr. Kelvin Drakemeyer from the Office of the Science and Technology Policy, the panel chair has addressed very important extract information from our study, which is talking about how this aerosol formed virus can form in the different pathways in terms of the transmission. So this is very important information for not only the academic, but also the government who are busy combating on the COVID-19 can be used for reference. And the Nature website has also reported a study putting into the information about is coronavirus airborne, and there are different evidences, and the study that we have done so far is a very important piece of this news report as well. So talking about the evidence that we provide from the Wuhan hospitals, how these results can help us understand better about this dangerous disease. Not only that, just 2 days ago, the Science website also reported our study about the ability of the coronavirus in this transmission, even by people's breathing or talking. And this is a very important evidence that we provide to our science community. Now here, the Science website has kept the important information about the personal protective equipment, the PPE of the medical staff that potentially can serve as a medium for transmitting the virus aerosol. Now, all this information together, I think is important for us to give a clear introduction and explanation of how we conducted the study. And in this 20 minutes to 25 minutes time, I will give the following list of the things in the presentation. I will explain the research team and how this study was formulated. And for some of the general audience and the general public, it might be important to understand more about the introduction for this SARS-CoV-2 aerosol. And the background of the aerosol transmission, that's very important background information for us to see, even as aerosol form, how this virus can be transmitted according to their own aerodynamic characteristics. So the last few points, we will focus on the study itself. I will explain the study design, the findings, the important ones, and also the conclusion and the discussion. In the end, we will recommend a few things that we see from the field that can help us understand better how to protect ourselves. Now, this study was formulated among five different institutes. Wuhan University, who actually inside the epicenter of the outbreak. So the State Key Laboratory of Virology, and in the Wuhan University, led by Professor Lan Ke and Professor Chen Yu, and myself is from the Hong Kong University of Science and Technology. I'm also from the Division of Environment and Sustainability. So our team here, also in the study, we designed the strategic sampling locations and also conducted the data analysis. From a few other institutes from Shanghai, the Environmental Monitoring Center, and also Fudan University on the School of Public Health. So we can see there are many important information that we can collect from this study using about the dissemination of information to the general audience through the angle for public health. So also this study is assisted with Professor Ho from the Chinese University of Hong Kong. So now this is the important background of the information for the virus aerosol transmission by different agencies. As we can see, during the outbreak of COVID-19, the different institutes, the different government agencies in different places in the world, they have a different understanding of this virus aerosol. They have also issued different guidelines how to combat on the transmission problems. So on January 31st, 2020, the Chinese CDC has released a guideline of the public protection of a pneumonia infected by SARS-CoV-2. So in this important guideline, for the first time, they confirm the transmission route of this virus, including aerosol transmission. Now, on March 27th, 2020, the WHO statement also mentioned about the mode of transmission being possibly transmitted between people through respiratory droplets and contact routes. But here, WHO is still very cautious. They haven't mentioned detailed about aerosol transmission possibility yet. On March 30th, 2020, the CDC from US, they have also issued a statement saying the virus is thought to spread mainly from person to person between people who are in close contact, also respiratory droplets produced with an infected person. So this is actually the common pathways of the aerosol virus transmission. And unfortunately, even at the moment, we don't have very solid direct evidence on aerosol transmission of this virus. And I guess this is also the main reason why different agencies, including WHO, has been very cautious about this mode of transmission. As you can see, understanding the aerosol transmission is very, very important for the prevention and control of COVID-19, and also for the effective blocking of transmission routes. And looking back about 17 years ago from SARS 2003, in Hong Kong actually, where I am at the moment, is actually the outbreak epicenter for the SARS. And both of the viruses, SARS and SARS-CoV-2, they attack the same receptor protein in our human body. And these two viruses have almost 80% of the gene sequence similarity. So there are many discussions and studies about the aerosol transmission for SARS in the early time. But the related research after SARS has been very scarce. We don't have so many evidences. Now, one study published by the Hong Kong University have shown the evidence of airborne transmission of SARS in the Hong Kong situation. So I guess the cautious thing we need to pay attention is when the epidemic outbreak of the disease spreads from person to person in larger scale and very rapidly, which is even beyond the normality of droplets or contact transmission pathways. We need to pay great attention of the possibility of aerosol transmission mechanism. Now, back to the virus aerosol itself, let's try to understand what is the virus aerosol we're talking about. If you look at this picture, showing in the air, when we talk about the particles or aerosols in the air, there are so many different possibilities. You can see that commonly when we say there's PM10, we refer particle size less than 10 micrometer. And if you go down smaller and smaller, you have a red cell, which is about average 7 micrometer. PM2.5 is much smaller, and the bacteria is even smaller. It's about 500 nanometer. And the virus we are talking about today it's super small. It's only a size around 100 and 120 nanometer. A few pictures just recently, we can see this TM picture of the new virus, the coronavirus, released by Chinese CDC, has shown the average size is about 80 to 120 nanometer. And then later on, the National Institute of Allergy and Infectious Diseases from US, they also published this TM image with a color enhanced. You can see the size is also about the 120 nanometer. So this is a very small size of the virus comparison with many other airborne particles like a PM10 or PM2.5 or even smaller. But why the size is so important? Why the aerodynamic property is so important for aerosol transmission pathway? We understand this one into two different angles. Now, number one, here we can see on the right-hand side, the size of the aerosol, the size of the particles that we inhale will determine where the particles will eventually deposit in our respiratory system. So aerosol initially, they are particles suspended in the air, but once we inhale them, once they go into the human respiratory system, if the size is very large, say above 20 micrometer, they usually cannot be easily inhaled into our nose. And if it's less than 10 micrometer, they are very easy to reach below the respiratory system. And if it is even smaller, say less than 2.5 micrometer, which is very important about the aerosol transmission now, they can easily penetrate our respiratory tract and reaches alveoli, which is very down here very deep into our lung system. Now, on February 3rd, a Nature study confirmed the high expression of SARS-CoV-2 receptor for this coronavirus on the epithelial cells. But unfortunately, this SARS-CoV-2 receptor protein is very abundant in our lower respiratory system, especially alveolar epithelial cells. So that means if the aerosol virus is very small, they can find a way to penetrate directly into our respiratory system and deposit right here in this region. And unfortunately, our alveolar epithelial cells also have very abundant expression of the SARS-CoV-2 receptor for the coronavirus. So you can have potentially high issues like the high deposition location and the high SARS-CoV-2 expression may cause very easy infection. So that's one way we look at this importance of aerodynamic property in terms of the size. The second thing about the size of aerosol is also very important. Now, this one is talking about how long they can stay in the air and how far they can travel when they are airborne. So that will determine the transmission capability of the virus aerosol. If you look at a few important pieces of information here, usually the size of the particles, if they're larger, usually they travel shorter distance due to its own gravity, and the shorter the resistance times they may have. Now, if the particles are very small, the farther they can travel, and the longer they can stay in the air, and the more easily they can get into our lower respiratory tract, as I just mentioned in the previous slide. If you do a very classical calculation about the setting velocity, and also assuming the particle size is 2.5 micrometer, and if you do the calculation, you will see that this size of the droplets and the particles, they can easily stay in the air for up to 2 hours time. Now, this is very important information as a background for us to understand how important the aerosol, the characteristics can be, especially for the virus aerosol. Now, we have a lot of questions to answer, but we don't answer them all, just to give us the information as a background. Just imagine if the virus, when they are airborne as aerosol form, if they stick together as a few, the virus, they can be very small in size. And each one is only 100 nanometer around. And they can aggregate as a form of a chain or the cluster of the virus is also small. And if it's the droplets coming out of the coughing or sneezing from the patients, or even by talking, the virus may be inside the liquid droplets. But these liquid droplets may evaporate. It's also possible, and we don't know the answer yet, that the virus may find their ways to stick onto existing particles. And these particles, if they're not so big, they can be transmitted in the air over different places. And their concentration may be high in the air, so the virus can find a lot of homes to stick into. So these are different senses that possibly the virus aerosol can exist. And we don't know the answer, but in this study, we will focus on another important angle. Here you can see from the Professor Lydia Barabia, from MIT, she has shown the high-speed camera, how this sneezing process, the coughing process, even the talking process can generate these large droplets. And during this process, actually, you can see a few different phenomena. Number one, the large droplets, usually by tens of micrometers, they can settle very fast, as just mentioned. They can usually travel by a distance up to 2 meters, which is also the current recommendation of the social distancing, about a few feet. So they are responsible for the droplet transmission, and you don't want to be close within this radius. Droplets can also fly in the air. They can, when they come out of the mouth, they can evaporate in terms of the water content, and the virus that can stay inside become the virus nuclear core, and usually the size about 1 micrometer. They can drift a much longer distance. And this is actually something we are worried about the aerosol transmission. And for sure, when we talk about the contact transmission, these kind of big droplets, eventually they will settle down. So the droplets with the virus, when they settle down on the surface of the objects, and if we, unfortunately, touch the surface by hand without washing, and you touch the hands by your mouth to your eyes, and this one become a secondary transmission by contact. So this is actually a very important background of how from the aerosol are formed, from the virus aerosol being formed to being transmitted and to be inhaled as the exposure to our human being. So this is the entire route. And we also need to understand the relationship between the virus aerosol and the room environment. And in this study, we have a few questions in mind. And you can see in this diagram, when we look at the droplet transmission, for sure we're looking at it very close to the patient. And the aerosol transmission, which is the focus of this study, we want to understand it better. To which extent the aerosol can exist in the air, and can we capture at all how many viruses inside the air in terms of the concentration, and what is the size of the aerosol that can determine two important things I just mentioned. The contact transmission is also possible. We have seen many studies, many reports showing they can find a lot of virus RNA on the surface of inside the toilet, in the patient rooms, in different locations, or even on the handle or the knobs of the door who is touched by the patients. So contact transmission is also important. And as in terms of the building control, the building environmental control to prevent the disease transmission, many of the buildings now, they have this the HVAC system, and these ones become a potential pathway for the viral aerosol transmission. We cannot answer all of the questions, but these are very open, very important things we need to address. For sure, as you can see from this the environment, the real world environment is way more complex than the simple diagram. There are different things we need to understand. Now, in terms of our study in Wuhan hospitals, what we actually have done is to look for three things. We want to understand the real world, the RNA concentration of the SARS-CoV-2 areas, in the medical staff areas, and the public areas. And what is the relationship between these concentration in the air versus the patient activities and the ventilation conditions of different places? The second thing is also important, and this is actually a very unique outcome and finding from the study, which is for the first time we identified the size distribution of this SARS-CoV-2 aerosol in the airborne form in a real-world situation. So number three, we look at the natural deposition rate of this virus aerosol in the patient room. And in this way, we can actually estimate what is the deposition rate per area and per time unit. So in that way, we can estimate the potential contact transmission in terms of this new coronavirus. We have designed the study very carefully in this strategy, and we have three types of sampling environment. The patient area, which is only accessed by the patient and medical staff. So inside this one, we include the hospitals from Renmin Hospital and Wuchan Hongfansan Hospital, which is renovated from the Hongshan Stadium. It's kind of a makeshift hospital to receive the live symptom patients. So we go to these different places, and inside here, our purpose is to study the effect of ventilation conditions in different setup of the hospital rooms, and what is the patient activities on the airborne this coronavirus concentration in the air. The second important area that we want to is the medical staff area. So these areas include but they are only exclusively accessed by the medical staff. I have to mention here that the general public and the patients won't be able to get inside. So whatever virus we can identify in this medical staff area, they are not primarily emitted or given by the patients. There must be some other transmission pathways. So that's a very important study objective. We want to understand the secondary transmission of this virus aerosol across different areas, and also the effect of disinfection measures on the airborne, the virus aerosol. Now, inside this medical staff area, we also had a chance to evaluate the airborne virus aerosol size distribution and the transmission mechanism of this virus, so in different places. The last one is about the public area. We look at the open space public area in Wuhan during the lockdown time, with the supermarkets and some outdoor places, schools, etc. And some places we even identify with crowd gathering too. So we want to see in this public area, is the air clean, or does the air contain certain level of the virus aerosol or not? So in this case, we hope to evaluate the effectiveness of the measures to prevent and control the COVID-19 spread. So these are the three important types of the sampling environment during the study. Inside the different environment, we also have three types of aerosol to collect. Number one, we call it the TSP, which is the total suspended particles. Now, no matter the size of the particle, we collect them all. And in this way, we can look at this total RNA concentration as an airborne form of this virus aerosol. The second thing is also important. We look at the size segregated samples in a way that we can separate the size of the aerosol in different stages, above 2.5 micrometer, between 1 and 2.5 micrometer, between 0.5 and 1 micrometer, between 0.25 and 500 micrometer. So this is actually the way we can separately look at the virus in which size ranges the virus can be dominant. Now, this is also important to know the transmission capability of the virus. When they stick onto or they aggregate as a single aerosol, what is the size range we reside? Number three, we also look at the deposition sampling. In a way, we have this natural deposition for 7 days in one ICU room. So nobody touches the sampling filter. We leave it still for 7 days. And inside the ICU room, we had patients. And after 7 days, we take the sample and look at this virus aerosol, how many inside, how many RNA we can collect. All of the filter samples, we used the gelatin filters for different reasons. And we have a long piece of the discussion about this sampling process in our publication. So you can feel free to go online and look into details. After the sampling we have done, in the analysis part, we look at the RNA copy number using digital PCR, bt-PCR, developed by the Wuhan University team. And we had a rigorous blank quality control as well. Okay, now a few pictures to understand the sampling process. And to be honest, this is a very dangerous and a very difficult sampling effort. And for the Renmin Hospital, for example, in the Wuhan University, so we went to ICU and CC rooms. So all of our sampling, the field crew members, they wear the very heavy protect cues, and they hold the samples in different locations by hours. And the Renmin University, Renmin Hospital, it is actually it's it's a grade A tertiary hospital in China. It's designated for the treatment of a severe symptom COVID-19 patients during the outbreak in Wuhan city. So you can see all of the ICU rooms and CC rooms, they are negative pressured with very high ventilation rate. The other type of the sampling for the patient areas in the Wuhan Fangcang Hospital. Now, this is relegated from Hongshan Stadium, as you can see from the news pieces. It's actually the very large open space with a big natural ventilation, and many of the beds are put together with the live symptom patients. So inside here, we collected the samples in different locations, and the most important one is actually the medical staff area, where they take off their protective equipment. And this Wuhan hospital is very representative of the makeshift field hospital. And as we can see from the news in many of the European countries, and even in many cities in the US, the government agencies have started to adopt this approach to concentrate to treat the live symptom, the patients, which turned out to be a very effective way. And here in this study, we look into details about different locations. Now, we also went to different outdoor sites, including the outpatient hall, some outside the hospital areas, and some of the supermarkets places. And in this way, we can identify what might be the concentration of this virus or aerosol in the air. Okay, here we come into the detailed explanation of the findings of the study. This is actually showing the virus aerosol concentration, TSB, inside the patient areas. So inside here, as you can see, in the majority of the places that we have seen, the concentration is very, very low. Sometimes undetectable, and sometimes it's below 5 copies per cubic meter of the air. So 5 copies per cubic meter of air. Now, we don't know are they safe or not, but across the different studies we have seen, this is a very low level. But in one location, in the mobile toilet room air, the samples have shown very high concentration of these RNA copies in the toilet room. So this is actually a very small mobile type of toilet, as you can see from the pictures. And we believe the virus may originate from the patient breathing during the use of the toilet, or they are from the splashing of the water and the creation of the liquid droplets from the toilet use. And as we can see in the studies from different agencies earlier in February, including US CDC and a few institutes in China, they have reported the detection of this SARS-CoV-2 RNA in the patient feces. So if the usage of this toilet room potentially can have this resuspension or re-aerosolization of this viral aerosol in the air, they probably can stay. So here in this study, once we identify the toilet room has very high concentration of the virus aerosol, we have suggested different measures, including provide the proper guidelines using the toilet. For example, we need to cover the lid for sure before you do any flushing. And it's also important to wear a mask if you do using the toilet in the public places, because we don't know who has been using the toilet in the public the places. It's very important to keep the disinfection process in place as well. Of course, increasing the ventilation in the toilets can be very important measure to prevent accumulated the virus aerosol in the air. And if you remember back in 23, the SARS period, one of the residential buildings in Hong Kong, once this virus aerosol from the toilet found their way out in the ventilation fan, they can actually be transmitted by air, and they can find a way to penetrate into different floors with the residents inside. So this is actually interesting, saying that the patient area, mostly they are very clean, but in the toilet room, the potential can be high. The reason for them to be clean is very high ventilation, and also the negative pressured measures can really protect the medical staff well in these locations. But a high concentration in the toilet room, for sure, because of this, the sources of the virus aerosol may reside in the feces of the patients, and also the bad ventilation inside the urinary inside the toilet room can be potentially the reason. The second important result I want to show is inside the medical staff area. This is also where our soldiers, we can call them soldiers during the outbreak, fighting with the disease. So this is where this medical staff, they take off their gowns, they protect their apparels, and we also where they have this intermediate space between the patient area and the public area. So here in this medical staff area, as we mentioned before, we assume there is no primary source of the virus. And if we identify the concentration of virus, they may come from somewhere that we need to identify, we need to know more. So from the study, as you can see, in the different locations, we have 3 different zones, A, C, B, and there is A. This is where the protective apparel removal room, where the patient, the medical staff, they remove the protective apparels. And in this, all of these, the red bars that we can see, they all happen in the Fanshan Hospital, which is a field hospital, as I just mentioned. So we detect very high levels in this medical staff area. So on average, the Fanshan Hospital has on average significantly higher numbers in terms of the viral RNA concentration compared with the Renmin Hospital, especially for the protective, the apparel removal room. What is the reason behind? We have 2 possible reasons behind to hypothesize. So number one, if you remember, the function of this kind of a room is to let the medical staff remove their protective apparel, to resuspension of the virus-laden aerosol from the surface of the apparel, which may be deposited during the working time inside the patient areas. And once they remove it, this aerosol, this virus-laden aerosol may gain its initial speed and it become airborne again. The second possibility is also the floor that has been deposited with the virus. It potentially can be a source of the virus-laden aerosol when people move around, and they can carry it across different areas from the patient to the medical staff area. And inside this medical staff area, when the staff is walking around, this dust on the floor that contain this virus may also be resuspended. So because of these important findings that we identify very high concentration in this medical staff area, we suggested that the medical staff should enter this room in order, in a way one by one or batch by batch to avoid cross-infection. Also, it's very important for them to, before they remove the protective apparel, a full body disinfection should be conducted. And from the results, it also seems very important. After removal, one more disinfection should be conducted in the room to clean up the surface. Now, very important, the side message, another remark about the study is after we identify such a high concentration levels in this medical staff area, we have reported to the government agency and also hospital authority. The further disinfection measures were taken, including increase the frequency of disinfection. We have a detailed explanation in the paper as well in terms of what has been done. Now, during the study, we have done this one in the middle of February during the outbreak. On the second batch of the sampling we have done on March the 2nd, we went to exactly the same place, same location, same sampling protocol. But the difference between here and the previous sample was, I just mentioned, there is a more rigorous sanitization process. And here, after 1 year again, we couldn't identify any airborne virus anymore. So here, the first batch study and the second batch study, you can see the big difference. And that actually really indicates how important it is to disinfect these rooms before the usage and in order to protect our medical staff. The last important, virus aerosol, airborne virus aerosol concentration report is from the public area, where we went to over 10 different locations in Wuhan in February. And in the public areas, you can see in the majority of the places, they are very low, below detection limit or even within 3 copies per cubic meter of air, which is relatively okay, safe. Now, when we look at other 2 locations, which have relatively higher concentration of the viral RNA concentration, they are from one supermarket and also from the outdoor of a living hospital. So this actually is the 2 locations where We don't have any information about if there were patients in the area, but we can hypothesize from the findings here that the areas that we have been to potentially we have seen a lot of a crowd coming in and out the hospital, coming in and out of the department store. And our sampling location is only 1 meter away from the pathway of the crowd. So once we identify this virus aerosol in the air in these locations, and we believe either the symptomatic the passengers in the crowd, and also the crowd gathering, when you have a lot of people together, they potentially can contribute to the elevated concentration of viral RNA in the air. Now, because of this, actually, we strongly recommend and suggest that the social distancing is very important, and it's very real, it's very serious, that even not inside the patient areas, because of this, some of the patients may know they have this, they are the carriers of the virus because they don't have any symptoms to show. So it's very important to do the social distancing and avoid the crowded gathering ourselves, because there may be symptomatic carriers in the crowd, who knows. It is also important to wear masks. Now, the reason for this one is not only to protect yourself, it's also to protect the other people. Because as you individuals, I don't think you know if you are a symptomatic carrier. You may not know because you don't have a fever, you don't have a coughing, but potentially you already carry the virus and you got the source of the virus in the crowded places. So wearing mask is a very important measure, not only to protect yourself, but also to protect other people. Number 3 is also the early detection, is by testing. It's very important. It's only through the early detection of this kind of symptomatic carriers, then you can avoid the crowd infection from the public. So that's a very important thing. You do the early detection, you do the early quarantine. In that way, we can cut this transmission pathway. So this is a very important finding of the 3 locations. Now, if we put all of the information together, 3 kinds of patient areas, in the medical staff areas and the public areas, you can see actually our public, our medical our patient areas, except for 1 or 2 exceptions. In general, the concentration is relatively low. So popular places, they are not so dangerous or with everywhere the virus. No, that's not the case. But in the medical staff area that you can see, it seems like there's a majority of these medical staff areas we can identify, we can detect the airborne virus. So that also indicates how important the work for the medical staff and how dangerous the working environment for the medical staff. The job of the government agents and the hospital authorities is how to better protect the medical staff, and in terms of doing the complete sanitization of these rooms, and also understand more the virus and take cautious measures. Okay, now second important types of aerosol we mentioned is about the deposition sample of the virus, the deposition. As I mentioned, how this deposition was done, we have 7 days continuously putting the sample on the one corner of the ICU room and another one in the other corner of the ICU room. Is about 2 to 3 meters away from the patient bed. And if you remember from the previous study, inside this ICU room, we couldn't identify any airborne samples. The concentration of this virus aerosol is very low in the airborne form. But still, after the study, after 7 days, we could still detect this virus aerosol deposit on the surface. And if we calculate this, how many copies of RNA on a certain area and over the time, we get 2 numbers in 2 different corners. As high as about 100 copies of the virus, 100 virus per square meter per hour. So that's a deposition rate. That's a deposition rate for the virus in such a clean ICU room. You can imagine in some areas where we could identify very high concentration, like 40 copies per cubic meter of the air is way much higher than this ICU room, this deposition rate may be much faster. So if you do the simple calculation, say on average, our body surface of an adult is about 1 to 1.5 meter to 2.0 square meter. Now, if you do the calculation, you can see after working in the ward for 5 hours, our medical staff, the surface of the gowns, the surface of the protective gears, we have over 1,000 virus copies. Over 1,000 virus copies. And if this medical staff, they move around, if they move around, they will carry this 1,000 virus copies around all the way into the medical staff area. So the very strict negative pressure conditions and a good ventilation in ICU, we can see they did remove the virus in the air that we couldn't detect in all, but still they cannot prevent the deposition of the virus on the surface. Now, this study is also very consistent with many other studies where they have shown doing the surface sample, they could identify the virus copies, very consistent from other studies. And it also reminds us that the floor cleaning and the surface cleaning become very important for the ICU rooms so as to prevent the carry of this virus by the medical staff from these patient areas to other locations. Okay, number 3 type of this aerosol sample is the most important one, which is the size of the SARS-CoV-2 aerosol. If you remember in the earlier slides, when I mentioned about how important the size can be, and we don't have any information because of the deposition location in the lung, because of the transmission capability, how long they stay and how far they travel. From the study, we identified the 3 locations. Now, 3 locations, if you see there's some general interesting point about this virus aerosol. These seem to be inside 2 size ranges. The orange box, here you can see, they are mostly below 1 micrometer. The other one is a green box. Actually, they are mostly above 2.5 micrometer. In 2 locations, we can identify both peaks. In most of the locations, we can see there is existence, sometimes very high, very distinct, below 1 micrometer. So we call them a submicron and the supermicron of these 2 different peaks. Now, these are all medical staff area, as I mentioned. This sampling site was only accessed by the medical staff, and there is no direct source of this SARS-CoV-2. But we can still detect this airborne virus, and where and why, and where do they come from? So here we have 2 important hypotheses about the mechanisms of this airborne transmission. A possible transmission of this submicron, the smaller size of the aerosol. The possible reason might be they may originally settle on the surface of the protective apparatus. That's just I mentioned from the deposition sample. And they can gain their initial velocity when the medical staff remove their gowns. And during this movement process, this small size aerosol may gain their initial speed, and they become airborne again and resuspended in the air. And due to the small size, submicron is a very small size. We mentioned that if it is a 2.5 micrometers, it can stay in the air for 2 hours. If it is a 1 micrometer, they can stay even longer, by hours. So this is very important thing to understand where this submicron, this small micron, the virus may come from, which is also a dangerous part of this virus. A second possible source for the larger size of this virus aerosol, above 2.5 micrometer. And we believe this is probably due to the floor deposition of the virus. Now that can be carried by the medical staff from these patient areas to the medical staff areas. And as they become resuspended when we walk around, and as this can resuspend this dust from the floor. And they don't stay long in the air, but they do present themselves in the air at the low concentrations, possibly. Okay, after we report the first sampling results to the hospital authority, we mentioned, because of this important finding that potentially the medical staff themselves are the carriers of this virus and become the secondary transmission pathway, the hospital authorities have done further cleaning process, which is to spray the disinfectant on the apparel completely before we take off, before medical staff take off their gears. And after implemented in this medical staff area, we couldn't detect any airborne virus anymore. So that's actually very, very important information for many hospital authorities to consider around the world, that potentially this deposition of the virus on these protective apparels and the remove of these apparels potentially can resuspend the virus on the surface. So we need to pay very, very important attention on this point. But in the end, when we say the conclusion and the discussion for this study, we have identified the submicron mode of the aerodynamic diameter of the virus, and potentially they can be very strong capacity in terms of the aerosol transmission. We also see a lot of hotspots with a high concentration of this virus RNA in different areas. And these areas do need improved disinfection measures. As I mentioned in the study, some of the measures did help reduce and even remove this existence of this airborne virus. And the deposition of the virus droplets or the virus aerosol may be potential important intermediate for the aerosol transmission. So the surface disinfection is very important, not only for the medical staff people, but also for our general public. If you go outside, you have to to the public places, either by going to the hospital or going to the supermarket, and you may touch different surfaces. And these surfaces may have the virus deposition. So it's very important for us to be cautious during the surface disinfection. Now, I want to also remind everyone that this study, we measure the viral RNA concentration. And we don't know, are they alive or are they dead? Are they still infectious? We can only see from the indirect evidence. For example, the study recently published in New England Journal of Medicine that has shown the laboratory generated the SARS-CoV-2 in aerosol form, generated from laboratory. They can stay in the air for a few hours. So that means if they are freshly emitted from the primary source for the virus aerosol, even they are in the air, they potentially can be alive for quite some time. And this becomes important, the secondary evidence to show how important the aerosol transmission pathway potentially can be. And we still have a lot of unknown about the virus. And I hope in this study, the findings can help us better understand the possibility of the aerosol transmission pathway. And also in the study, we have recommended a few important measures from the Wuhan experience. For example, the ventilation and the sterilization of toilets, which is a potential source of this virus. It's important for the ventilation and sanitization. The second thing is the personal protective measures for the general public, like wearing mask and avoiding busy crowd. It can help us reduce the risk of virus-laden aerosol exposure. As we can see, next to the hospital, even in the open areas, and also in some of the entrance of the busy crowd in the supermarket, we could also identify the airborne virus. So we need to protect ourselves by wearing masks and try as much as possible avoid the busy crowd. And number three, the effective sanitization of high-risk area in the hospitals that can help limit the transmission of airborne virus and protect the medical staff as well. Number four, the effectiveness of the naturally ventilated large stadium, like the Fangsan Hospital we mentioned in this study, which is renovated from a big stadium. It can really limit the aerosol transmission because of the large ventilation, open space. So it's important as a reference if the other countries have the need to build their own field hospitals. This is a very important message. Number five, the surface sanitization of the apparel before they are taken off to help reduce the potential infection risk of the medical staff. This is also important. So these are all the important recommendations from the Wuhan experience. So my presentation is over, but in the end, I want to bring up this two pictures. One was the time Well, the first time the man went to the moon and did the scientific experiments on the moon with all of the dangers. The second picture is our field team inside the Wuhan hospital with the sampling process. And you can see they are all very, very serious petechials, and they are also in a very dangerous environment. Now, I was asked by many friends, it's why such a dangerous environment, still we want to go to do experiments. I would respond with a sentence I can remember. Because our ultimate pursuit to expand the boundaries of human knowledge, it led us to the moon and also give us the power, give us the courage to conquer the field of the unknown. I'm Ning from the Hong Kong University of Science and Technology, and thanks for your time.